This video is going to be on the new methylene blue stain. You will perform this stain out in practice if ever you want to perform a manual reticulocyte count or identify Heinz bodies. Fortunately, our newer hematology analyzers give you a reticulocyte count, so most likely out in practice, you'll just be using this to identify Heinz bodies. You perform this stain by first adding equal parts of the patient's EDTA whole blood and the new methylene blue into a sample tube. And then you swirl these components to make a nice mixture of the red cells and the stain. And you let this sample sit for 15 minutes at room temperature. And it's during this time that the stain is making the RNA aggregate, and the stain's also binding to the hemoglobin. After the 15 minutes is up, you make a regular blood smear just like you guys performed in lab, and you let the smear dry. Then you evaluate it with the microscope, and you'll use Kohler illumination. Okay, so now let me show you some pictures of some reticulocytes and Heinz bodies. First, here's a picture of an aggregate reticulocyte. You can see the nice blue clumpy aggregates within the red blood cell. These reticulocytes are seen in the peripheral blood of all species except the horse. Cats have a second reticulocyte called the punctate reticulocyte, and it's just little pinpoint dots of reticulum. Heinz bodies look like blue circles within the red blood cells or sticking out from the red blood cell.